Hello Year 9. This lesson is all about discovering DNA, the recipe for life. You will need your Life's Like That booklets. You need to turn to page 36, please. So normally at the start of this lesson, I would be bringing in some cake to share with you. And the whole purpose of bringing this in is as an example, an analogy, okay, to our recipes. So if you look at the two imaginary cakes, because I'm afraid there is no cake that we can share with you during COVID at the moment, we have got on the left a Tesco strawberry and clotted cream flavoured Swiss roll made with buttercream, and on the right a double chocolate Swiss roll made with buttercream. So when we look at these two items, I want you to see if you can identify the similarities between them and the differences between them, first of all. So if we look at similarities, they're both cake, they both come from Tesco, they both serve 8 people, they're both suitable for vegetarians, they're both a similar kind of packaging, they both have a swirl in the middle, they both have a filling, they're both tasty. I'm sure you can think of lots of other ones. How are they different? Well, the one on the left is made with a strawberry and clotted cream flavoured vanilla sponge. The one on the right is a chocolate flavoured sponge with a chocolate buttercream inside. How else are they different? They have slightly different packaging in that one is purple and one is pink. And you could say they have different tastes to them. And I'm sure you can think of some others. So why are they different? This is where we come to our analogy and why I'm talking about Swiss rolls first thing in the morning. So they are different because of their ingredients. So if we were able to turn the packaging over, we'd be able to see the ingredients that are found in the strawberry flavoured one and in the double chocolate Swiss roll. So how does this relate to us? Well, if you look around you in the class, there are similarities between all of you. You're all human. There are no aliens in this class, okay? So you've all hopefully got all the things that a human should have. But you've also got differences. So your differences might be your hair colour, your eye colour, your height, shape of your facial features like your nose, your ears. And the reason you are different is because you have different recipes. Where are these recipes found? Well, they're found inside every single cell of your body. So if we take a skin cell, a hair cell, a muscle cell, a brain cell, every cell that has a nucleus, so remember our red blood cells don't have a nucleus, every cell that has a nucleus will have DNA inside it. And that DNA will be exactly the same in every cell in your body. So answer the questions at the top of the page. Ask the teacher to pause the video so that you can do so. Including these two questions at the top. Do you think humans have different recipes and that's why they're different? And where might we find this recipe? Obviously in our nucleus of every cell. So what is this DNA? Well, it actually comes from a very long word, the DNA, from deoxyribonucleic acid. So there we have the D, the N, and the A. So you don't need to remember the big long word, okay, but you know it as DNA. So if you look at our little picture down the bottom here, we can see a typical animal cell. It's got a cell membrane, it's got a nucleus, it's got cytoplasm, and in the nucleus you can see these little X-shaped structures. They are called chromosomes. So when we take one chromosome and start to unravel it, we see this very familiar shape, which is called a double helix. And this is the shape that DNA has. It has 2 backbones going down the side. If you can imagine taking this DNA and twisting it to unravel it, it looks a little bit like a ladder when it's unraveled, and it does do this during when the cells are making copies of themselves. So it becomes like a ladder, and across the middle of the ladder are the rungs, the things that you put your feet on, okay, as you're walking up the ladder, and the 2 sides, okay, of the ladder are like the backbone. So along the backbone, we have a sugar and phosphate backbone, so alternating. So you'll see this in a bit more detail later on. And across the middle, the rungs, these are what we call the bases. And there are actually only 4 of these different types of chemicals making up your DNA. But it's the order of these, and there are millions of them, okay, to give everybody that unique code, that unique pattern that makes them. So the 4 bases are called adenine, and we remember them with letters A. Thymine is T, cytosine is C, and guanine is G. And you should be able to see in this diagram that the purple and the green molecules join together, that's the adenine and thymine, and the pink and the blue molecules join together, that's the cytosine and guanine. And Mr. Hancock may be able to bring out for you a model of the DNA so that you can see its 3D structure. It looks a bit like a spiral staircase. A section of this DNA is known as a gene that we can see here. And it's these genes that make us different. So that's what we're going to be looking at. Now, at the very bottom of page 36, you should be able to see an image where there is a picture on the left-hand side of some of these X-shaped chromosomes all matched up, and a picture on the right-hand side. Now, we call these diagrams a karyotype, okay? And scientists carry out karyotyping, okay, for various different reasons. So it may be that when a lady is pregnant, she may have a test called an amniocentesis test. And an amniocentesis test is to find out if there are any problems with the baby. So what they can do is they take a needle guided by ultrasound and they pass it under local anaesthetic through the lady's belly button very carefully, because we don't want to skewer the baby that's inside the uterus. And they take out some of the fluid that the baby is floating in. And this fluid contains some of the baby's cells, because babies are constantly shedding some of their skin cells whilst they're in the womb, just like we do, okay? So you are sat here at the moment, bits of your skin cells will be falling off. That's what makes all the dust that collects around the place, okay? So we can take some of those cells and we can take the nucleus out of the cell and we can analyse the chromosomes. So this is really important for scientists to be able to do this, to identify if there are any issues. And what they do is they match them up as pairs. Now you have a pair of chromosomes, okay, for every pair that humans have. And we have 23 pairs of chromosomes, okay? Everybody has 23 pairs of chromosomes. So we pair them up. The biggest start first at number 1, and we arrange them in order until we get to the last pair. And we always find that this last 23rd pair, depending on what sex the baby is, will be slightly different. So we've got, if the pair are the same, are exactly the same, we know that the child will be female. But if the pair are different, if the last pair don't seem to match, then that tells us they're a male. And we call these sex chromosomes the X and the Y chromosomes. So a double X, two the same, is our female, and an X with a little Y chromosome is our male. Now I've ordered the Chromebooks for today. Hopefully somebody has gone down, a couple of pupils have gone down to go and collect them, because I wanted you to have a go at doing exactly the same as what the scientists do, matching the chromosomes up in a karyotype. Now traditionally we used to do this as a cut and paste activity. So I would normally be giving you a sheet of paper and you would have to cut all of the chromosomes up and you would have to pair them together. But technology, we can do this now electronically. So on my PowerPoint, I'm just going to take you to the next page. There is a really good website, okay, from a university in Utah in America. And if you go to this web address, so once you've got yourself logged on, okay, to the Chromebooks. So I'm sure Sir will pause the video, or whoever is taking the lesson will pause the video for you. Type in this web address once you get logged on, and it should take you to this page. I want you to leave the hints off if you can for me, okay? Because I want you to try and do this without the help, okay, to begin with. And all you do on the website is you click on any one of these chromosomes and you drag it and match it to its matching pair. So I just want you to spend a few minutes, okay, dragging those chromosomes, okay, to the correct places to match them up. So pause the video at this point. So if you completed it, hopefully your completed karyotype looks like this. Now, when we say that the chromosomes are exact matching pairs, you can see that their shape sometimes isn't the same. So for example, chromosome pair number 8, this one looks a bit bendy. Because chromosomes are quite flexible in their nature, we don't expect them to be dead straight. So that is what makes it even more challenging to try and match up your pairs when you are doing this task. We have our 23 pairs for this person's DNA. Can you tell me what sex their DNA is, or what sex this person is that the DNA has come from? So you should be able to tell, because this last matching pair isn't exactly the same. This is a male and they have X and Y chromosomes. So on page 38, can you answer the questions? Okay. And the little black and white bands, so you can see them quite clearly here in this chromosome pair number 1. They are exactly the same place on the chromosomes. These are the genes. Nothing to do with your Levi's or anything to do with denim. Genes spelt with a G, and it's the genes that determine our features and our characteristics. What is really amazing is that scientists started mapping all of the genes on human chromosomes when I was in school. And they completed this, I think, just after I finished university. And basically they've mapped the whole of the human genome, okay? In other words, they know exactly which chromosomes, okay, certain genes are for different characteristics. So the genes that control your hair colour, your eye colour, your skin colour, but also not just physical features, your metabolism, how your body works, how fast your body metabolizes certain chemicals. And also they've identified the genes that cause us problems where we have inherited disorders. So things like cystic fibrosis or Down syndrome. So Down syndrome, there is an extra chromosome 21 just here. Okay, so remember that one of these chromosome pairs has always come from mum's egg, and one has always come from dad's sperm. So there's mixing up of your DNA from your parents, from your mum and dad, okay? And that's for every single one of these 23 pairs of chromosomes you've got mixing up. Every time your parents decide perhaps to have another child, a sibling, if you've got brothers or sisters, your brothers and sisters may have some genes that are the same as you, which explains why they look similar, but some of their genes may be different. Because remember, you're only getting half of the DNA from dad's sperm and half from mum's egg. And that is what makes everybody unique. So if you look halfway down on page 38, you should be able to see, so these are the answers for the questions at the top, a diagram that looks like this. So I've called this task halving and doubling to explain where that DNA comes from that we've just been talking about. So in the mother's egg-making cell, so we know that's in the ovary because we've studied the sex organs recently, there are 46 chromosomes. In the father's sperm-making cell, so this is in the testes where the sperm are made, there are also 46 chromosomes. Now what has to happen is in the ovary and the testes, the sex cells, or a posh word for these sex cells is gametes. The gametes have to be made. That is our egg and our sperm. There's the word gametes there. And we want the egg and the sperm to have half the number of chromosomes. We call this the haploid number. So if you remember half, so lots of key vocabulary we're learning today. The number at the beginning in the mother's egg-making cell and father's sperm-making cell is called the diploid number. Di meaning 2, okay, or double. So what has to happen is a type of cell division that we call meiosis. And meiosis is a cell division where the 46 chromosomes copy themselves first of all, so they would become 92. And then they split once, so that we have 2 cells with 46. And then they split again, so that we have 4 cells with 23. So I've just shown 1 cell here. So this is happening in the ovaries right now and in the testes right now to make eggs that have half the number of chromosomes and to make sperm that have half the number of chromosomes. So when they join at fertilization, key word that we learnt about in the last few lessons when we're looking at development of the fetus, the 23 and 23 join together, so you get this mixing up of DNA of mum and dad, half from mum's egg, half from dad's sperm, to make a full set of 46. So we say this is our diploid number again. This fertilized egg cell is known as the zygote. And what will happen is that will then start to divide to become the embryo. So if you remember when we went back to what was happening after fertilization in the egg tube, that fertilized egg cell was copying itself. It started from 1 cell, divided to become 2, 2 divided to become 4, 4 divided to become 8, to 16, to 32. And obviously these cells are all joined together in a ball, okay, to make up that what we call early embryo. It's called a blastocyst. And it's at this point, when it's around about 32 cells big, the ball, that the cells are all exactly the same. They all look like typical animal cells, and it buries itself into the uterus lining. At that stage then, those cells then start to specialize. So we've got a few questions that I need you to answer. So pause the video, see if you can answer the questions at the bottom of page 38. And here are the answers. So how many chromosomes are there in an egg and a sperm cell? There are 23 in each. What is the scientific term for a sex cell such as a sperm or egg? It is a gamete. When the sperm fertilizes the egg, how many chromosomes are in the fertilized egg cell? 46, or we can sometimes write it as 23 pairs. What's the scientific term for the fertilized egg cell? It is a zygote. Turn over the page to page 39, and I'm hoping that Mr. Hancock has got you some coloured pencils that you can borrow, or that you have some of your own. Now, we're going to look at the structure of DNA in a bit more detail. So we know that this is called a double helix, this shape, okay, and obviously it is a 3D shape, and it is made up of sections a bit like a jigsaw, okay, called nucleotides. So to show you what a nucleotide is, if we have the DNA model, you'll be able to see that there is a base, which is one of the rungs that go across the middle, the ladder. There is what we call a 5-carbon sugar, and we call a 5-sided shape a pentagon. So this 5-carbon sugar is sometimes called a pentose sugar. So the prefix there, pent, meaning 5. And it's attached to a molecule called a phosphate group, okay? So these separate nucleotides, there are 4 different types of them, okay? And the reason there are 4 different types is each one has a different base. So this base may be adenine, or it may be thymine, or it may be cytosine, or it may be guanine. And these bases, these chemicals, okay, all have different shapes. And as I said, it's a bit like a jigsaw, so the shape of the base will fit exactly. C and G will fit together, so we can see the blue and the red joining together along this spiral helix, and A and T join together. So what I would like you to do is I want you to colour in your diagrams. I want you to colour in your, first of all, the example of the nucleotide, this one over here. Then I want you to take 4 different colours. Now, if you haven't got the same colours as shown in the PowerPoint, don't worry, as long as you remember which colours need to join together. So remember A and T and C and G. And I want you to colour over your chain and then answer the question for, is there a rule for the way they pair up? So remember, A always pairs with T and C always pairs with G. So you can see the DNA unravelled, because I did say earlier this does unwind when it's making a copy of itself. You can see the nucleotide first of all here with our phosphate, our pentose sugar, our 5-carbon sugar, and our base. And what they've done here is they've given the base, the cytosine C, a little pointy arrow shape, because its opposite one that it joins with has got the reverse shape so that they fit together like a jigsaw. And these little dotted lines between them are representing the hydrogen bonds. So they join together by hydrogen bonds. A and T, we can see A has got this kind of curved shape, and T corresponding, so it fits like a jigsaw, has got a kind of concave shape so that the A can join together. And we can see the hydrogen bonds joining A and T. And what you should notice is that C and G always have 3 hydrogen bonds between them, and A and T always have 2 hydrogen bonds between them. So you can colour code this diagram on page 39, but there is a slightly bigger version on page 40 to show you what DNA looks like. And this is the one that I would like you to colour code. And you can see our sugar phosphate backbones where it's unwound, and you can see them joining together with the different shapes. And this is important, okay, because you will also see this model, this one that's been unwound here, at the back of my classroom, hanging up as a big banner. And I made this many, many years ago, and I wanted to actually attach them to wire, so I could take the wire and twist it to make our double helix, so it could be hanging from the ceiling like a big spiral. But the wire was going to be so expensive, okay, more expensive than the model, that if Mr. Hancock has been able to show you, okay, that model was about, in its 20 odd pounds, and the wire was going to cost about 40 odd pounds. So it didn't make sense to be spending that kind of money on the wire to make my model, okay? And that should take you to the end of this lesson, okay? So we'll have a look at this code and what this code means and how it translates to all your features and how you inherit your features a little bit later on. As a little additional task, if you have finished this task, you can turn to page 44. And on page 44, there are some diagrams. I'll see if I can just take this to page 44. So discard those annotations. And page 44, you've got these boxes. I'm going to come off of them very quickly because I don't want to give you the answers. See if you can put the missing words into those boxes for me for the last task. And you will be having a go, you'll see over the page in a later lesson, at creating your own DNA kind of sequence and your own cartoon character based on a DNA sequence. Okay, thank you, Year 9.